For gene expression studies, we need to obtain the complementary DNA sequence or cDNA sequence for a gene. First, let's take a glance on the section of the genomic region, transcripts, and product. We can see that insulin gene has 4 mRNA transcripts variants. If we are evaluating the insulin gene expression on the gene level in a novel cell, then we have to carefully choose which one of these 4 mRNA transcripts we need to use to design a primer. The trick is, we have to design a primer with an exon region that is shared by all the 4 transcripts, because we don't know which transcript is expressed in the novel cells. For example, if the novel cells express this transcript, and we designed a primer in this region, then we will never get a PCR product because the region we are targeting doesn't exist in our novel cells' mRNA transcript. Therefore, it's better to choose the first transcript because it shares all the exons with the other transcripts. Go to NCBI reference sequence. From mRNA and the protein section, select gene bank of the first transcript variant. This page displays all the information related to this sequence. Let's take the first 2 exons. The location of the first exon is from 1 to 42, while exon 2 from 43 to 246 base. You see that there is no difference between the end position of the first exon and the start position of the second exon, because the mRNA transcript variant has no introns. The mRNA sequence is actually cDNA sequence. Another thing to know, when we design a PCR primer, it's better to design inside the CDS region or the coding region, because this is the actual region of DNA that is translated to form protein. This is the location of the CDS sequence. To obtain this sequence in FASTA format, scroll up and click on FASTA. Now the FASTA format is the format we are going to use to design a PCR primer using a primer design tool such as a Primer 3. From change region shown, you can specify which sequence you want to show. For example, if I want to show only the sequence of exon 1, specify the position of the first exon here, which is from 1 to 42. Click Update. To retrieve the reverse complementary sequence for exon 1, go to Customize View, enable Show Reverse Complement, and click Update. Another way to retrieve a specific exon sequence is as follows. Go to Insulin Gene page, and from here, select Gene Table. These tables below show you the start position number for each exon in each mRNA transcript variant. Also, it shows you the intron size. In transcript variant 1, click on exon 1 location, and here we obtain the exon 1 sequence. You can also retrieve the coding region sequence. Click on coding of the second exon, and here it is. In the next lecture, we're going to use the FASTA format sequence of a transcript variant 1 to design a PCR primer using a Primer 3 program.